Well, I certainly am going to be talking about biospecimens, but even more fundamental than that, I'm going to be talking about standards in this process. Biomarkers are measurable signals that reflect an underlying biological state. Now, most of these are measured from biological samples taken from patients. I'm a pathologist, and pathology is the medical discipline in which the vast majority of biomarker analysis for clinical use and patient management is performed. Biomarkers are indispensable to the practice of medicine as we know it, and as evidenced by the fact that 80% or more of the objective evidence in data, information in a patient's medical record comes from the pathology laboratory. Biomarker uses include detecting disease before the appearance of symptoms, making correct diagnoses, selecting the right therapies, detecting complications, making predictions about and monitoring the course of disease, and predicting the response to specific treatments. As diseases are more specifically characterized on a pathological level and classified on a molecular level, and therapies are developed against specific molecular targets, the practice of precision medicine, as was mentioned, becomes possible, but not without the development of new precision biomarkers. This is needed to guide both drug development and for complex disease management in this new era. In short, new biomarkers will be essential to realize the promise of precision medicine for the future. And because biomarkers are guideposts for critical decision-making in medicine, their measurement must be reproducibly accurate and consistent, and reliably reflect the biological state of the individual, not giving random, biased, or false information to the decision maker. Their measurement also needs to be technically reproducible, independent of who is making the measurement or in what pathology laboratory they are being measured. While the causes of irreproducibility are diverse, many are related to the lack of unifying, broadly implemented standards across the biomarker continuum, from discovery right through implementation into the clinical system. Such standards would dramatically reduce the irreproducibility and increase the efficiency of translation from phase to phase in development, save incalculable amounts of money, and increase stakeholder confidence in biomarker utility and investment worthiness. In biomarker development, as in all of biomedicine, the best standards are themselves evidence-based. An overarching fundamental problem that currently exists in biomarker development is the lack of focus or effort spent on the development of the data on which to create evidence-based standards for biomarker development. Finally, there is a complete absence of a unifying standards framework within which to apply and monitor standards, working backward from those standards which would be implemented clinically right back to the first principle of biomarker discovery. The NBDA intends to meet all of these challenges, but it expects the task of establishing standards for biomarker development to be complex, and we further expect that the complexity will increase as technologies for measuring biomarkers continue to become more powerful, faster, better, cheaper, as they have in the past decades, and will continue to be in the future. This is now an era in which omics-based technologies that can measure hundreds or thousands of molecules of the same class simultaneously are being applied to biomarker development. These technologies, in turn, require sophisticated information technology systems to analyze the massive amounts of data that are generated, and equally complex are the multifaceted biomarkers that are comprised of signatures of molecular entities of different types, or even of combination of molecular-based biomarkers and imaging-based biomarkers. These are integrated with computational algorithms, which at the end of the day become essentially the biomarker itself, a highly complex approach. All of these cutting-edge approaches require the application of fit-for-purpose standards to ensure reproducibility and applicability. So across the board, the need for standards at all levels is great, and the need for a workable, scientifically sound standards framework is great. Meeting these needs will require cross-sector engagement of all stakeholders, as Dr. Barker mentioned, and this is exactly the approach we intend to take. Standards have enabled breakthroughs and served as the foundation of progress in global implementation for many industries. For example, transformative innovation in the form of the ATM card, the semiconductor, Wi-Fi, the internet, and others required standards to accomplish. We have that same opportunity now to advance medicine in a transformative fashion through the creation of evidence-based fit-for-purpose standards for biospecimens, the biomarkers that are measured in them, and the development of new technologies for the measurement purpose. At present, not all stakeholders have fully recognized the value of biomarkers, and irreproducible results, high failure rates, and staggering costs have undoubtedly contributed to the heavy loss of confidence. We intend to turn this around. We believe that a systems-based approach to standards will support the production of credible, reproducible, and translatable outcomes in biomarker development, and robust biomarkers will, in turn, create and enable a new era of medicine for patients. And in the end, this is our aim at MBDA, to change the medicine of the future for the benefit of patients. Thank you, Anne.